All right, home stretch, 2 more modules, and the last one's short, so let's get going here. So hopefully the last 2 modules started to have a bit of a similar flavor, and you can see the process we go through. Different problems, different setups, but the basic ideas are always the same. So now we're going to look at phylogenetic models, right? So again, we're going to have a slightly different structure to the problem, but we're going to end up going through the same process. So before jumping into the modeling problem, let's think a little bit about what data we have available. So in phylogenetics, there's a pretty big distinction between whether you have bulk or single cell data. So first of all, bulk. There's really 2 major ways to use bulk data to try to build sort of phylogenies over clones. The simplest and earliest and probably maybe most commonly used is to use the presence or absence of mutations in a sample to try to build a tree, right? Once you have the presence or absence of a mutation in a sample, this looks a lot like a standard phylogenetic problem where your species are samples and your characters are the mutations, right? And you can sort of do all the obvious things like parsimony or Bayesian methods. So there's an alternative set of methods which try to do deconvolution, like we saw in Module 2. They try to work out the mixture structure of each sample while simultaneously reconstructing the tree, right? Or not simultaneous, but while reconstructing the tree. And within this subclass, there's 2 schools of thought. Again, one school is that we do the deconvolution first. So, for example, do exactly what we did in Module 2, and then you take the cellular prevalences and try to build a tree. The other school of thought is that you just try to do the deconvolution and tree building together. There's the same trade-offs between these 2 schools of thought as with the copy number approaches where we segment first or don't. It's usually a trade-off between computational complexity versus feeding information back into the other step. What I should say about these approaches, pretty much the core assumption.
One question is this not also, what is about iterative approaches? When you do the first step, are you kind of iterative?
Sorry, say it again.
So what is if you go to the first step, are you like iteratively? So kind of
then you're kind of in the second class already, right? That's almost like an EM algorithm. So usually it's a pretty hard call. We get the prevalences and we go from there. We don't change them. What I should say, actually, I'll turn to the questions first, because I think this one may elicit some strong opinions. What is the issue of doing classical phylogenetic inference with bulk data? Are samples really like species? Yeah, the analogy I think of is you want to build trees with species, but a sample is like just going into a forest and throwing all the genomes into a blender and then trying to build a phylogeny between different forests across the world. The question is, what does that mean, right? And the biggest problem is the mixture issue, right? That is the issue. And what that means is just because we see 2 mutations in a sample doesn't actually mean that they're in the same clone, right? So what's our tree? So that's sort of that is the benefit of the deconvolution approach, right? Because we're going to try to solve the mixture problem simultaneously, right? The issue with the deconvolution methods are these ones that try to jointly build a tree and look at sample mixtures. What do they have to assume to be able to build a tree? Has anyone ever looked at these? The essential assumption is that the higher the cellular prevalence of a mutation, the higher it is up the tree, right? That was the picture I drew in module 2, right? Assuming our cellular prevalences are completely accurate, why is that still a problem?
Something about mutation that is constant over time.
What happens if you have a copy number event
Copy number varies.
that wipes out the mutation? Yeah, exactly. You have lost mutations, and we'll talk about that later in this module. But again, there's trade-offs. So you may ask why we are actually going to do this. We are going to build a classic phylogeny over samples, and you may ask why we're doing that. Because it seems dumb. Well, one thing is you can actually handle loss in that case, whereas you can't do it in the other one. We'll come back to that in a bit.
Sorry, can you say you want to do this? So the first bullet point
Yep.
is you are actually, I mean, you want to apply standard phylogenetic methods to multiple samples, for example, to many tumors from many different patients.
No, same patient.
So
Different places. Yeah, sorry. This is the same setup as module, the deconvolution thing. We always assume that within a patient. Phylogenies between patients, I think Nico may have some ideas on this he's going to talk about next. But I mean, you have to take a very different definition. And what we mean, though, here is a set of clones that are evolutionarily related, right? And that. Oh, a sample is just different biopsies. So you have metastases or whatever, right? Different parts. Yeah. I hope that was clear in module 2. That's what I meant by sample.
Sorry, I was just not.
No, not you, but I was just thinking I actually never explicitly stated that. So that's a good point. So the other type of data that we have now is single cell data. And this maps much better onto phylogenetics as sort of we classically know it, because cells are species, or they're individuals within a population if you're thinking about the coalescent, right? Now, why is single cell not the answer to every problem? I mean, there's always availability of data sets. There's way more bulk data than there is single cell, so it's interesting. But single cell data right now is noisy. That's the answer. What is the problem with single cell? It's always noise, right? So you may think, well, what if we wanted to look at SNVs as markers in single cells? How feasible is that? We've already discussed this. If you have 0.1x coverage, not very feasible. CNVs are a potential event we could use because they are pretty easy to detect if we use binned read counts like we talked about. And actually, you can get very accurate CNV profiles from single cell data on certain platforms, right? But I don't know who has thought about this, but modeling the evolution of copy number on a tree is actually surprisingly challenging. And it's just because you have a lot of convergence. So if you think about a single bin, if we're sort of looking at independent bins, we'll have a lot of different ways to get it to a specific copy number, right? We have multiple changes, this, that, and the other. And we don't actually have a very intuitive model of how copy number actually works and what causes the changes. And so if you think about sort of classical phylogenetics of nucleotide variation, you typically have some sort of transition model between different nucleotides that's pretty easy to interpret, but there's not the equivalent in copy number space. So you can try CNVs, but I'm going to say it's challenging. One thing that seems promising in the single cell space is to actually look at the change points associated with any specific copy number event. In some sense, these should be quite unique, assuming that your bin data is small enough, right? So though, if you look at a single locus, it may be affected by multiple different copy number events as we go down the phylogeny. The actual change points of each of those events will typically be different, right? So there's a wrinkle with this idea.
It's probably the resolution of the bins, is it?
That's one. So if your bins are big because you have low coverage data, many change points will be in the bin. There's another wrinkle here, which is how do we get the copy number profiles from the different cells? You can basically run the model we just talked about or something similar, and you can do that on each cell, right? If you never run an HMM, this may not be surprising, but where it actually decides to start a segment is quite variable, like within sort of a couple positions. So now all of a sudden you have a bunch of different cells, and if you look at them, you can pretty much eyeball it and say, that is the same change point. But because you've analyzed them independently, they're going to be slightly different. So there's no clear event you can say, this is what I'll use to model with, right? So now you need a new model to solve that problem. I think people are working on it. It's an interesting problem, but there are issues with change points.
Isn't there also a problem if you do like a joint inference on the plasma cells, on the breakpoints, and then use that to pull the tree from the joint inference profiles? You get some sort of bias. If you have a bias in
A little bit, because you're assuming a clustering structure. You're basically assuming the clade structure already, right? Yeah, it's tricky. So maybe you want to build the tree and do segment, but that's going to be hard, right? This is always the trade-off in modeling, right? We can back to the pre-processing versus joints question.
What about the calculations between two pair of cells, like the evolutionary distances and passing down a little bit and so on?
That's people are working on that, and again, it's sort of, this doesn't really matter whether we're talking about probabilistic modeling or doing distance-based methods or anything. You still need to come up with a good good data representation, right? And sort of address any of these issues. Distance-based methods will still struggle a bit if you have really noisy data or you can't get the change points aligned and things like that. So many of the problems you face in the probabilistic version of phylogenetics, you'll hit in the distance-based method. The key feature of the distance-based methods will be scalability. If you have 100,000 cells, nobody's ever really tried to do Bayesian inference on phylogenies of 100,000 species. I mean, it's hard. Pardon me?
You can group them somehow.
It's kind of cheating, though. Well, it's easier with species than with cells, though, right? I guess. But yes, it's a hard problem. So distance-based methods tend to scale a bit better, right? They tend to be faster, like neighbor join is. Well, you still need the quadratic penalty for building the distance matrix, but no.
Sorry, I wanted to ask, so when you say change points are possible, so does that mean, so what do you actually mean by saying character? So you want to say treat them as
Yeah.
similar?
Yeah, we just treat them as IID.
But aren't they like pairwise related?
They are. So that's the approximation. They are pairwise related, and you may have to phase two different pairs.
But you don't know which pair with which.
You can model this out if you think about it. It's hard, but it's doable. If you ignore it, there's going to be an issue. There's an approximation, but is it going to cause is it actually going to really affect things? Open question. I don't know why the magnet stopped working on this. But yeah, there is the pairwise issue. I don't want to go too deeply down this rabbit hole. My main point to this slide is how to do phylogenetics with single cell is still pretty wide open. We have this whole issue of how do we even represent the data before we can think about anything else.
This pairwise section, is that, do you mean because you have like, when you have one break and another break there, but it's not special?
Yeah, I'll draw the picture. If you have a copy number change, you have,
These two ends are like,
yeah,
but it's
and then you
not
can start,
spatially, so it's not like,
pardon me?
I mean, it's not, position is not related, it's just the effect. Like,
You'll get two events on a branch instead of one. So in some ways, actually, one thing I thought is maybe the worst case, you just double your branch lengths, and you're going to do it for all of them.
The problem is that like, there's a quadratic number of pairs.
Pardon me? No, but you don't need, so what I'm saying is you ignore the pairing constraint. Worst case, you just accidentally double your branch lengths, but you do it across all the branches.
So the thing is, when you want to consider pairs, then you end up in a similar problem as you were mentioning, as how do I acquire a CV? There's multiple ways of getting there. It's the same CV, right? Or the same set of CVs. You could first
Right, but now we have
identify something and then delete something in the middle, or the other way around, right? And the same thing is with the pairs.
Not necessarily, because then we can just think about which pairs do we see in cells together, and if we see enough, we can sort of hope to see that. We don't actually need necessarily a model. We just need to pair them. We don't need a model of how the pairs evolve. That's why CNV is harder. Yeah. That's like this is a full-on research problem, I will say. It's a little bit, let's say, advanced. Point is that single-cell data representation is even just a key issue. Okay, I'm going to forget about single-cell data right now, and I'm going to go back to bulk sequencing. And the classic phylogenetic setup, I've sort of alluded to this already, is we have a data matrix, an observation matrix, where we have m rows corresponding to species, and we have n columns corresponding to features or characters, right? And what we want to do is we want to infer this evolutionary tree relating species, tau, where we have a set of edges and a set of vertices. In some cases, we'll have branch lengths. And we have this thing where we can have either a rooted tree, where there's a well-defined ancestor, or an unrooted tree, where we know the relationship, but we don't necessarily know where the ancestral point is, right? So I think a question here in cancer is, should we use rooted or unrooted trees? Which one?
Because you also know that's
Unrooted or rooted? Sorry. Yeah, and I would agree. I think we always use rooted because we know the ancestral sequence, right? We always know that these cells come from a normal cell, right? So we do know the root. And this is actually a case where things get easier in this domain because we do know the ancestor. So the basic ingredient of a phylogenetic model is to define transition probabilities. So formally, or sort of informally, what we want to say is, given that your ancestor had some character, right? Say it's nucleotides and it had a C, what is the probability that its child all of a sudden has a T, right? And that's the transition probability matrix. So the classic approach to do this is to define what's called the rate matrix. Here, the interpretation of the entries is it's the instantaneous rate of transitioning from character I to character J, right? And then to compute the actual transition matrix, you exponentiate this matrix multiplied by the branch length. If you don't have branch lengths then things are a bit easier. You can just directly define the transition matrix. Now, given the transition probabilities, what we'd like to then do is compute the probability of the data. The problem with phylogenetics is the notation is a little tedious, but the ideas are pretty simple. We're going to have a lot of things here, but the idea is pretty easy. So we have xv, which are the value of the characters at the leaf nodes. Y is the value of the characters at the internal nodes. L of tau is the set of leaf nodes. Big I of tau is the internal nodes, excluding the root. Tau of v is a subtree rooted at v. So, just so everyone's on the same page with this. If this is my whole tree, then, and this is V, then tau of V is this little tree here, right? That's the tree rooted at V. Rho of V is your parent, so if I'm here, that's rho of V. Gamma of V is the set of children, so that's these guys. And then R is our root node.
Character is the way you define
It just means this species 1 has an A here at this position, species 2 has a T there, etc. It's, yeah. When I talk about the value of the internal nodes, it's a little bit more obscure because we don't typically observe the internal nodes. Here we're doing the standard probabilistic modeling thing where we assume we know the value of these hidden things in order to get to a tractable way to compute a probability. But once you have all this notation, then you can write down the probability of your data, given a tree topology, a transition matrix, and here we're going to assume you have a labeling of all the internal nodes, right? And then you can write this 100 different ways. This is just to show you a basic one, and this is a recursive definition. In practice, we don't know those internal labels, right? So again, I emphasize we've done the trick of introducing these things to come up with a probability model. But what we're ultimately going to do is marginalize those variables from the model. That is, we're going to sum over all possible labelings of these hidden internal nodes, right? So the sort of magical thing, or maybe not so surprising after seeing forwards backwards for HMMs, is that you can do the summation over all possible labels of the internal nodes efficiently. Naively, it looks like it's going to take quadratic time. If you do it efficiently, you can do it, sorry, not quadratic, it's going to be exponential in the size of your character set. But you can do it more efficiently in quadratic time. And the basic idea is just to come up with another set of recursions that look a whole lot like the forward-backward. The main difference is we don't do a two-pass thing. We only do a single pass up the tree. And I won't go, you can look into most books, but this is called the Felsenstein pruning algorithm. In a lot of the literature, it's a dynamic programming algorithm. The key thing about this recursion is that we need to initialize it, and what we do is we initialize the values at the leaf nodes to 1 or 0, depending on whether that state matches what we actually observe or not. And then we recursively move up the tree from leaves to internal all the way to the root, applying this equation. This is kind of quick.
In case we have instead of A, C, D, E,
Yeah.
we will have an N, we will have 1, 1, 1, 1 in the teams.
Pardon me?
If you have errors in the teams, instead of having
Oh yeah, we can have.
A or C or C, you have N or these characters.
Then you just put a uniform probability. Yeah. Actually, that's a point that we will come back to later in this, is that you don't need to have a 1-0 initialization to this recursion. It can actually be a probability of being in these states, and we're going to leverage that later when we build a model. So the whole point of the pruning algorithm is it gives us an efficient way to compute the likelihood of the data given the tree, right? To specify a Bayesian model, we're going to need priors. So we're going to need a prior on the tree. If we have branch lengths, we'll need a prior for that. And then you potentially will have a prior over any parameters that dictate your transition matrix P, right? So the biggest issue is that phylogenetic inference is hard. There are roughly order n factorial trees for rooted trees with n species, right? So if you have more than, say, 10 species, that's a big number. Trying to do sort of things like map estimation is hard because this is a discrete space. We talked about this in module 1, right? So all you can really do, you can do a bit better, but really all you can do is brute force enumeration of every single tree. But once you've done that, you have computed the posterior, basically. That is your normalization. That's the exact amount of computational effort it takes. So this is one of these rare instances where it's no worse to do MCMC than to do map inference. And that is why you will see the vast majority of Bayesian work in this field is based on MCMC methods. Beyond just uncertainty quantification, it's because it is no harder. All right, so now that's the review. Let's turn to the model we will build. And we are going to consider a probabilistic model for mutation loss, right? So we're going to do sort of the thing that I said was bad and build phylogenies from bulk sequence data. But we're going to try to do it in a way that's not so terrible. What we're going to do is assume that we have these M samples from the same patient with SNVs, right? And roughly, we want to have, roughly the entries of this matrix should be the presence or absence of the mutations. But actually what we will encode is the probability that a mutation is present in the sample, right? We'll come back to that in a second. So obviously, obviously this is a challenging problem because samples are mixtures of clones, but there's also this issue that mutations may be lost due to CNVs that overlap the mutation. And if this isn't obvious to you, here's sort of a simple schematic. So we sort of have our mutation tree, and we have this orange mutation that maybe it originated in the root node. Somewhere along the way, we just lost the chromosome that it was on, so it's a lost mutation, right? This is the thing that breaks the deconvolution methods that try to build trees, right? Because you can no longer rely on the assumption that cellular prevalence is an increasing function of tree depth. So the model assumptions we're going to make here, I'm fast tracking a bit the model building process by this point, but the assumptions we're going to make here are that mutations originate at most once on the tree, right? So this is the infinite sites assumption. And that's saying that we don't expect a point on the genome to get mutated twice because mutation rates are low and the genome's a big place. What we will allow, though, is mutations to be lost after they are acquired, right? And this can happen multiple times, right? And this is actually a good reflection of the way copy number works. It's quite common to have the same locus affected by multiple independent copy number events. The other thing we're going to assume here is that mutations evolve independently on the tree. If they're really close together, this is not ideal. But all we're basically saying is we can write the likelihood as a product of likelihoods for each mutation. You can relax that a bit, but it gets hard. One thing, and this comes back to the point about the 0-1 probabilities for initializing the recursion, we're not going to assume that we can perfectly observe the data, right? So we have noisy measures of presence-absence. So our input matrix won't actually be a 0-1 character matrix. It'll be a matrix of probabilities of presence, and in fact, more formally, clonal presence of the mutation. Now, the reason we need this, one is sequence coverage, tumor content, those things. So even if a mutation is in a sample, there's always a chance we just don't sequence it, right? The other issue is the clonal mixture thing. And what we hope is the fact that its clonal presence will sidestep this. And I don't have this. The basic intuition, if I know two mutations were clonal, that is, in the most dominant clone of the sample, then I know they're together, right? And in fact, if you could say confidently that two mutations were above a cellular prevalence of 0.5, they must co-occur in a cell, right? There may be another cell that only has one of them if one's at a higher cellular prevalence, but you can at least say they co-occur. So this, to some extent, if we could do it well, would sidestep the mixture issue. So we're going to have a pre-processing step here where we compute this probability of clonal presence. The notation's a little bit different, but this shouldn't shock you. This is just the exact same rationale we used in Module 2. We're going to introduce the number of copies that are mutated at a position, the total number of copies, and the tumor content and the sequence error. And then we're going to have this probability of having a read with a mutation. This should look familiar. And it's going to look like this. So this is basically a simplified version of what we worked up to in the previous example. We've set cellular prevalence to 1 here because we're assuming it's clonal. You can actually do something that's a bit less dumb, and you can integrate over cellular prevalence from 0.5 to 1. But regardless, you can get probabilities in this way. So, and then we do the classic thing. We model the data as binomial, and we obtain the probability of presence by marginalizing over this unknown state, which is the number of mutated things. This is all sort of exactly the same rationale we went through previously. And again, we also use copy number data to inform how we do that. So we end up with a slightly non-standard version of the pruning algorithm in this problem. The main issue is the single origin assumption creates dependencies among the state parts of the tree. If I say that a mutation originates here, I know that it's definitely absent there, right? So to work out what those, to work out the pruning algorithm for this, we need a bit of notation.
What was the rationale for the single origin assumption?
It's infinite sites. You don't get it.
Is it makes it more feasible, or?
What? No, it's to model the fact we don't want mutations popping up all over the tree. It seems wildly implausible that I independently acquire the same mutation in 3 different species, or 3 different clones, right? That's what we want to avoid. There's a more general probabilistic model called the stochastic Dolo process that encodes all of this stuff. This is kind of this is a very simplified version of that. But the key assumption is we want a mutation to only appear once, but then once it appears, it can get lost. It can be lost in the tree. So we'll need a variable pi L, which is the probability a mutation is lost along a branch. We're going to have this probability ZV equals 0 is the probability a mutation is absent at node V. So somewhere in the background, I'm introducing an indicator variable on whether a node has the mutation or not. And I'll have this probability ZV equals 1, the probability it's present. And so what we're going to compute is this thing Q, the probability a mutation is present at node V, given all possible histories of mutation loss on the subtree, right? And it looks a lot like the Felsenstein pruning recursion. And this is our initial condition. So V is in the leaf nodes, and this is for the internal nodes. Basically, both of these break down into 2 parts. The event that we lose it at that node, more correctly, we lose it on the branch above that node, but we just say at that node, and the probability that we don't lose the mutation at that node, right? And so for the leaf nodes, we weight those by the observed probabilities that we have. And then for the internal nodes, we perform this sort of recursion using Q, right?
The assumption is still we know all the internal nodes, right?
No, the recursion lets us marginalize the internal nodes. The only assumption we're making here is we know that what this is computing is the probability a mutation is present. I'm tired at this point. We're marginalizing over the states. That's the whole point of the recursion. We're not assuming we have the labels of the internal nodes. Because basically, this is summing over both possible labels. Yeah, right? Because we have a 0-1 problem. It's both labels. All right. So let's assume, now we make an assumption of a thing we need to know that we don't. Let's assume we know the node W, or where a mutation originates. Then the probability of the data given the tree in that node is given by this value, where it's the probability of being present at that node over all possible loss histories times the probability all the leaves outside that subtree are 0, right? So if we know that the mutation originates here, anything else on the rest of the tree cannot have it. That's the second part of that. Now we do, hopefully by now, the standard thing, and we sum out all possible positions of this mutation origin. And then we need a prior for that. We pick the uniform prior over nodes on the tree. Here, just there's an absence of any preference on where a mutation should originate. Then we now appeal to the idea that mutations are independent, and we rewrite the product of all our data. Or sorry, we rewrite the probability of all our data as the product over each data point. So it's all looking pretty clear. So inference is the question. The paper where we actually published this model, we had a small number of samples. And typically if one does multi-region sequencing, more than 8 or 9 samples is not terrifically common. You can get more, but it's just not. So if you are in that regime, then it becomes feasible to enumerate every single tree, compute the probabilities, and find the map estimator, right? The nice thing too is you can compute the probabilities independently for each tree, so you just spread this out massively. Even with massive parallel computation, the strategy caps out at about 10 or 11 samples. But if you're in the small regime, you can do the simple thing. So that's what we did. And we also optimized the value of the probability of loss for each tree then. And doing that, then you can come up with your map estimators of the tree and loss probabilities. Okay, this will break with more than 10 samples, then you need to go to MCMC. We didn't do that because we didn't want to write an MCMC sampler for tree inference. It's hard.
Just a question, is this complexity comes from the number of possible trees? Because you said Felsenstein is kind of quadratic in some way.
So Felsenstein lets you score one tree efficiently,
Yeah.
but you have
So
n factorial trees to score.
And Felsenstein is a square in number of characters.
I may have it, it should be quadratic in the number of characters because you have to sum over.
Characters is those mutations you have, so.
Sorry, no, characters means what is the states that any given site can take. So for us it's 0, 1, we only have 2. For nucleotides it's 4, amino acids 20, so forth. Yeah, so the more you have, the harder it gets.
I guess it's correct in the number of internal nodes, because you store internal nodes when you go from bottom to top.
No, it should be linear in nodes because
all right.
it's quadratic in states. It should be linear in nodes. You can only do it once for each node. The exponential comes from enumerating all possible combinations. The naive version comes from enumerating all configurations. All right. So once you have your point estimates, you can do the same thing you do with HMMs and essentially run something that looks like Viterbi. You essentially replace the summations in the Felsenstein pruning by maximization steps. Once you do this, you can compute the most probable sequence of presence values along the tree under the constraint that things only appear once. And that gives you a labeling that you can then infer where a mutation originates and also where it's lost. So that gives you something like this tree. Here, because we don't have branch lengths in the model, what we do is we scale the branches by the number of mutations assigned to them. So the length of this branch means we have about 3,000 mutations acquired in the ancestor. We also have losses annotated with these numbers here. So we lose, say, 110 on this branch. And then because we can map and we're able to do this because we can do that mapping of mutations back to, or the origin of mutations back to a point on the tree. Right? So we can say that 3,000 mutations originated here under our map estimate. But sort of clear. And then you have this relationship among samples. So the one question is, does this idea even make sense and work? And how would one validate it? So this will be the one time I talk a little bit about model validation. And this is not rigorous, this is just intuitive. But if we have a mutation we predict to be lost, right, we would hope there's a copy number event coincident or overlapping that position that explains that it's lost. If we don't see any copy number change, why is it lost, right? That's sort of the question. That's the point of this. Maybe I'll walk you guys through this. What this plot is showing is allele-specific copy number profiles for each of the samples that we built the last tree with, right? Now, we're looking at a small region, and within this region, there's a set of mutations we predict to be lost. They're predicted to be lost in this sample and that sample, right? And in the other samples, we're plotting the presence of them with these little sticks at the bottom. So you can see in the lost samples, we don't see the mutations, and in the other ones, we do, right? The point of the yellow highlight here is we have a very focal allele-specific deletion that perfectly overlaps all these losses, right? It's some evidence that the predicted losses are valid. It's not great, but that is evidence. Does everyone understand what I'm saying with this? All right. Of course, you can't talk about why a model is good without talking about why it doesn't work, and this is where you need to have a critical eye. So this model fails gloriously in some situations. Here is one where, again, it's the same sort of plot. All of the mutations we're plotting are ones that are predicted to be lost somewhere in some sample, right? And so what you're seeing is these mutations are spread uniformly across the genome. If you actually look at the counts, the number of lost mutations in this particular patient is massively more than any other patient. You could say something really interesting is happening here, or you could say, okay, the model's broken. Why? And the answer is the model's broken here. And to understand why it's failing, we need to think through a few cases here. So what are we really doing when we use this type of presence-absence information to build a tree? So here's a simple phylogeny. What we're basically doing is counting the number of mutations that are shared between two samples and putting the ones with the most mutations shared together. So that's how we know, for example, S1 and S2 belong together and not with S3. This is kind of the optimal case for building clone phylogenies from samples, because no sample is a mixture of clones from totally different parts of the phylogeny, right? There may be two different clones here, but they're all sort of from one part of the tree, and they're not represented anywhere else, so it's not going to badly break things. What we won't be able to do is resolve that these two clones are different with this approach, but we won't sort of get a grossly incorrect topology. So this is the perfect case. Once you introduce mutation loss, things get a little bit harder. And the basic issue here is if you play the counting game, these two share one mutation, but S1 and S3 also share one mutation. So it's unclear. With such a small number of mutations, this seems ambiguous, but if you have lots and lots of other ones, then this becomes unambiguous. S1 and S2 actually will share a lot of others. So as long as we can account for the fact that loss sometimes happens, it should be a big deal, right? This is the case that breaks the model. And it's when you have two clones from completely different parts of the phylogeny in the same sample. And again, if you play your accounting game here, it's unclear if S2 belongs with S1 or S2 belongs with S3, right? The problem the model has that we just discussed, it isn't aware of this. It doesn't try to do clonal mixtures. So the only sort of knob it has to adjust to data that doesn't fit is loss. So if you end up with a sample that's an even mixture of two clones from different parts of the tree, it's just going to have to use a bunch of losses to explain it. Good. So that's the end of this module. I just want to summarize the last little bit there and say it's important to actually look at the output of the model critically. It's good to look for validation that it's working. It's also good to look at cases when it fails and really understand how it's failing and sort of how to diagnose that it's failing. One of the nice things about this model is when it does fail, it is so obvious because there are so many predicted losses, right? And if you actually look at the paper this was published in, we didn't even try to use it on patients where we saw this, we just did single cell sequencing. All right, do you guys want to take a short break?